The University of Connecticut says vaping causes DNA damage, but their study is full of holes. According to the University of Connecticut, their new study says that there's evidence that electronic cigarettes are potentially as harmful as tobacco cigarettes. If you've read the full study, which I have, and not just the article and the abstract that UCONN released, you'll see plenty of holes in their research. And how about this quote that I pulled directly from the intro of the study? Additives in recent e-cigarettes make the vapor less harsh and allow more rapid delivery of nicotine to the brain, fostering use and increasing chances of addiction. So that's not true. Anyway, so this study was performed by chemists using a 3D printed testing device. They found that e-juices that had nicotine caused as much DNA damage as unfiltered cigarettes, and that non-nicotine e-juice caused as much damage as filtered cigarettes. The researchers believe that the non-nicotine e-juice causes DNA damage because there are still many chemical additives in the flavors of the e-juice. The lead author of the study, Kartik Kedamisetti, a postdoctoral researcher at UCONN, said that the amount of DNA damage caused depends on several factors, such as how much vapor is inhaled, the different additives in the e-juice, if the e-juice has nicotine, and other factors. He concluded that based on their study, they are certain that e-cigarettes have the potential to cause as much DNA damage as unfiltered cigarettes. So let's dig into the study. First, let's talk about the 3D printed device that they invented. This is a new electro-screening device developed in their lab. It was designed to detect genotoxicity, which basically means damage to genetic material such as DNA. The device pushes liquid samples across microwells that contain human enzymes and DNA samples. The camera detects light when reactions are generated. The more common testing method for genotoxicity is by using bioassays, which are more comprehensive and trustworthy, but also much more costly, which is why UCONN created this new device. UCONN themselves say this device is an initial screening tool. The researchers used artificial puffs by drawing vapor from electronic cigarettes through a tube that had a cotton plug. The samples were then taken from the cotton plug and tested on the DNA and enzymes. And it has been pointed out online that this is a major flaw because DNA and enzyme mixture is not representative of the cell, tissues, or organs in a living organism, which also means that it's not representative of the effects that you might have from vaping. They tested both e-cigarettes and tobacco cigarettes for 3 known carcinogens at 20 puffs, 60 puffs, and 100 puffs. They found that as the puffs increase, so did the likelihood of DNA damage. The 3 carcinogens that they tested were benzoapyrene, NNK, which is a nicotine-derived nitrosamine ketone, and these have some long names, so 4-methylnitrosamino, 1,3-pyridyl, 1-butanone, that's one of them, and the other one was N-nitrosanicotine. So benzoapyrene is a product of incomplete combustion and is found wherever organic materials are burned. It can be found in coal tar, tobacco smoke, and grilled meats. Since nothing is being burned when you vape, it's not likely to be found in vapor. Both of the NNKs, or the NNN, these are nitrated derivatives of nicotine. It's been shown in other studies that they're not present in vapor in significant amounts. And also, most NNKs originate from nicotine burning. Those carcinogens require burning, which leads me to believe that the researchers were not testing the e-juice at realistic or normal conditions. And if you read the full study, there's no mention at all of what device they used, what e-liquid they used, the PG-VG ratio of the e-juice, how much nicotine was in the samples, the wattage or temperature of the device, what coils or wicking material were used, or even puff duration. And these are pretty significant facts to leave out. One of the things that bothers me most about this study is that they are testing 2 different things, the genotoxicity of e-cigarettes and also this new device that has just been invented. Well, 3 things actually, because they also tested wastewater. If you look closely, you'll notice that this study is mostly shilling this new testing technology, not actually thoroughly testing electronic cigarettes. And this genotoxicity study should have been tested using well-established methods of testing, such as bioassays. And if they wanted to test the validity of the new 3D printed device, they should have compared the results of the new screening method to the more comprehensive and trustworthy bioassay. And the study was also not performed in vivo, or in a whole living organism, such as a human. It was an in vitro test, which means that it was performed on tissue extract. Even in vivo tests on animals are more reliable in finding results that relate to humans than in vitro tests. And in vitro tests are also mostly performed using cancerous cell lines that already have abnormal function. And it's also known that in absence of human physical activity, the results of an in vitro study can be misinterpreted. All of these issues are especially true in toxicology studies like this one. I've had a viewer of mine specifically call me out on his YouTube channel for ignoring negative vape studies on this show, which isn't true at all because I talk about negative studies all the time, but usually to disprove them. But the thing is, you get studies like this where something is wrong. There were no details at all of the vape gear that they tested and at what temperatures, durations, or anything. Without knowing these things, this study is completely useless. For all we know, the researchers might have been feeding their machine dry hits, which is actually very common in e-cig studies. Have you ever seen a legitimate scientific paper leave out so many details? There's probably a reason why they left all of this out. And that's why I don't talk about every negative study I see, because in a lot of cases, it's just not worth my time. So anyway, is DNA damage something to worry about? Maybe, but this study doesn't prove that conclusion.